Pleasure now to introduce Dr. Andrew Chan. Dr. Chan is the Daniel K. Podolsky Professor of Medicine at Harvard Medical School, Professor of Immunology and Infectious Disease at the Harvard T.H. Chan School of Public Health, the Chief of Clinical and Translational Epidemiology at Massachusetts General, and the Director of Epidemiology at the MGH Cancer Center. Among his numerous awards, he is the team leader for the Stand-Up for Gastric Cancer Interception team, the co-team lead for the Cancer Grand Challenges Early Onset Cancer Team, an NCI Outstanding Investigator, an American Cancer Society Research Professor, and a member of the American Society for Clinical Investigation and the Association of American Physicians. We're very delighted to welcome Dr. Chan.
Thanks, Robert. This has been a fantastic conference, and I want to thank Juha and Robert and all the organizers for having this summit. It's really been a terrific opportunity to connect with the gastric cancer community, and so I'm very grateful and honored to be here. So my task today, I think, is to give you an update on the work that our Stand Up to Cancer team has been doing related to early detection and interception of gastric cancer. So I think this talk will be very much shifting gears from what we've been talking about yesterday, but I hope will give us some sense of where the science is headed with respect to early detection. So back in 2020, I came to the first summit, and that was when our stand-up team first launched. And so it was a great opportunity to kind of lay out our plans for what we were going to do. So it's nice to be able to have this opportunity to provide an update. So I think back in 2020, I did emphasize one of the major goals of our team, and I think the goal of most of the people in the room today is really to shift the curve when it comes to gastric cancer. I think we're all very aware of these very significant and alarming survival curves with respect to gastric cancer, where we know that when people are diagnosed early, here in the blue with stage 1 disease, they do quite well, whereas people that are diagnosed with stage 2, 3, and 4 don't do as well. And certainly we know most of the people in the United States present with stage 3 and 4 disease. So the real purpose of our team was to actually shift the curve and make sure that we could move more people from those later stage curves to stage 1 disease. So how do we do that? And I think we've all are familiar with this very clear opportunity for prevention with respect to our understanding of the progression of gastric cancer from normal gastric mucosa through the various steps of gastritis, atrophic gastritis, intestinal metaplasia, adenoma, dysplasia, and intestinal type gastric cancer. And if we classify the various phases of the disease, we can see quite clearly that most of the time when we think about this disease, we're focused here on the cancer stage. But we really wanted on our stand-up team to be able to focus on earlier stages of disease. We've been largely in this summit talking about this predominant phase or progression of disease in gastric cancer, but we also very much wanted to focus on the other major subtype of gastric cancer, which is diffuse type gastric cancer, which does sometimes go through this sort of cascade of molecular events, but also is quite clearly on a separate path, particularly in the hereditary form of gastric cancer in individuals that have hereditary predisposition like mutations in CDH1. And as many of you know, one of the major challenges in this type of gastric cancer, particularly in the CDH1 population, is that there are no real clear precursor lesions. So there really is a challenge with respect to how do we find ways to successfully intercept and prevent diffuse type gastric cancer. So as you all know, the clinical practice in the US at least is largely focused on patients with cancer, and our goal was really to try to shift clinical practice to earlier stage disease. How were we going to do that? Well, our original purpose in assembling our stand-up team was to really focus on two major areas of early detection and interception. One was the development of blood-based biomarkers, and then the other was the development of advanced imaging approaches. And advanced imaging approaches is an area that our team has been actually quite successful in. We've actually been doing very interesting studies relating to molecular imaging of gastric cancer, which has showed some very promising results. Unfortunately, I won't have time to go through those results, but suffice it to say that a major emphasis of our team has been on molecular imaging, and we're looking forward to continuing work in that area. So we recently have extended our studies of molecular imaging into doing fluorescent endoscopy using a molecular imaging probe, which we're quite excited about. For the purposes of today, though, I want to focus on blood-based biomarkers, and in particular, a really interesting scientific area of blood-based biomarkers, which is the development of a new mode of detection, which is to detect a specific protein in the blood. Our stand-up team, though, really wanted to look at the landscape of blood-based biomarkers. So we did evaluate a variety of different molecular and circulating products of gastric cancer as potential blood-based biomarkers. So over the course of the last 3 years, we've done studies looking at circulating tumor cells, cell-free DNA, and exosomes, and had various results that were quite intriguing, but nothing really moved the needle when it came to early detection. But one area that we found to be particularly interesting was to look at the proteome, and that's where I'm going to focus my talk today, with the idea that we could shift clinical practice to, again, earlier detection and allow us to more successfully intercept gastric cancer at a stage where it's potentially curable. So I first want to recognize that this has definitely been a team effort. The Stand Up to Cancer network has been a tremendous opportunity for us to assemble an international group of investigators that have been really focused on gastric cancer. And I think, as we mentioned yesterday, there's been a lot of momentum, I think, with respect to the gastric cancer community, which I think we've been able to really capitalize upon through the funding from Stand Up to Cancer. So in 2020, Stand Up to Cancer really, I think, shifted a lot of their interest into early detection, and we're really grateful that one of the areas they wanted to focus on was gastric cancer, which was tremendously understudied. So I had the privilege of leading this team along with my co-lead, Sandra Riom at Columbia University. But this was really an international effort. So we also had team leads from Samsung Medical Center, in Seoul, Korea, with Dr. Ji-Yeon Lee. We also had Yang-Hee Woo, who's here in the audience from City of Hope, and also participation from the NCI, Jeremy Davis. In addition, we've also been guided by our patient advocates. I think all of you hopefully had a chance to hear Aki yesterday afternoon. Aki Smith, who is the director of the Stomach Cancer Awareness Network and Hope for Stomach Cancer, has been really an invaluable source of inspiration for our team, and also provided some really essential input in terms of how we would progress the science into our patients. And then finally, I want to recognize Marty Taylor. Now, Marty was our team member at Mass General. He's a gastrointestinal pathologist and has really spearheaded a lot of our work in terms of blood-based biomarkers. And he recently departed just down the road to Brown University. But he's been really a linchpin in trying to put together our early detection blood-based biomarker work and really has spearheaded the work that I'm going to present today. So I'd like to first talk about the science behind what we're doing. And I think what we had a very brief discussion about yesterday was the idea that we could think about multi-cancer early detection tests as potentially an option for early detection. But as many of you know, the potential options at this point for MCED testing are largely based on looking at things like cellular DNA or exosomes or fragments of DNA or the methylome. And it's been difficult to be able to really look at MCED testing for early detection because it has not so much been tumor-informed. It's really been based upon thinking about just what are the byproducts of cancer that could be circulating in the bloodstream. So in contrast, what we tried to do in our team was also not only look at those sort of agnostic markers that could be early detection biomarkers, but also try to do something more hypothesis-based. And that's why we really started to focus specifically on line 1. So I do want to take a step back, realizing that not a lot of people are familiar with line 1, and so I want to talk a little bit about what line 1 is as a specific area of interest for our team and why it leads to potentially such interesting capabilities potentially for early detection. So line 1 stands for long interspersed elements in the genome. So many of you know much of our genome is comprised of repeats. The most common repeats that people are familiar with are microsatellite instability repeats that are seen throughout the genome. But there's also the major other type of repeat in our genome is known as line 1, or these long interspersed elements that are throughout our genome. There's over 500,000 copies of these line 1s throughout our genome. And it's really something that we think of teleologically as being related to almost what was seen evolutionarily as sort of viral transposons. These are repeats that are sort of inserted throughout the genome through sort of a viral retrotransposon method. In the vast majority of our cells, these line 1 repeats are actually not expressed in the genome. They're repressed by many different cellular mechanisms. And in normal tissue and normal somatic cells, line 1 is repressed and not expressed. But there are situations where line 1 is actually expressed, and that is where line 1 potentially becomes a promoter of cancer. So just to look a little bit at the life cycle of line 1, line 1 is sort of a retrotransposon that's in the genome, and it has two major functions. It's the only transposon that actually has a function in terms of protein coding. It encodes two different proteins. One is ORF1, and the other one is ORF2. And why is it important? Well, ORF1, this protein, is a specific RNA binding protein that is pervasively expressed in cancer. In addition, ORF2 is another protein that's synthesized that actually has retroviral reverse transcriptase function and a nuclease function that also may be implicated in the pathophysiology of cancer as well as many other diseases. So what happens in a situation where line 1 is de-repressed is that the RNA transcript actually goes into the cytosol, and in combination with the proteins that it encodes, specifically ORF1p and ORF2p, synthesizes additional products which can then go back into the nucleus and then insert itself using reverse transcriptase from the ORF2p protein back into the genome. And the problem is that when line 1 is reinserted into various parts of the genome, there is the opportunity for genetic mutations to occur and for a lot of genomic instability events to occur that leads to development of cancer. In addition, we know that there are functions of this line 1 RNA in the cytosol. So this protein complex that line 1 forms can form RNA and DNA hybrids that actually causes inflammation in a pro-inflammatory state and also can activate innate immunity. So our team actually provided or manufactured this slide to kind of give people some of the insight into line 1 because it's not a very straightforward pathway.
The human genome contains approximately 500,000 copies of a virus-like element known as line 1. In certain disease states, line 1 escapes repression and generates RNA molecules that migrate to the cytosol, encoding proteins that can transfer line 1 RNA back to the nucleus, where through reverse transcription, they can create and insert a new line 1 DNA copy into the genome, causing mutations. Reverse transcription can also happen in the cytoplasm, where it creates RNA-DNA hybrids, aberrant products, which can trigger inflammation. The detailed structure of the line 1 reverse transcriptase enables development of specific inhibitors that may become effective therapies for autoimmunity, cancer, neurodegeneration, and other diseases.
So that gives you a sense of the biology. So thinking now about this biology, it provides a unique opportunity for us to think about this as a target for both prevention of gastric cancer, but also as a potential target for early detection. So the first step in terms of thinking about it as a biomarker is whether it's expressed and how it's expressed in various cancers, and specifically, is it overexpressed in gastric cancer? So this was work that was done in various cohorts of cancer patients that we've assembled at Mass General to look at the expression of line 1 in various types of cancer. And suffice it to say, there is significant overexpression of the line 1 protein as detected by immunohistochemistry in GI tract cancers, in particular colon, gastric, and esophageal cancer, as well as ovarian cancer. And this protein, this line 1 protein, also ORF1p protein, is not expressed in normal tissue. So you see very clear differentiation between normal and tissue. And this is an example in colon cancer. And we also found this to be the case in gastric cancer. So in a cohort of 51 gastric cancer cases that we assembled through our Stand Up to Cancer biobank, we found that there was significant overexpression of line 1 in gastric cancer as compared to normal. And it's also very pervasive in early gastroesophageal precursors. So we also saw overexpression of this ORF1p protein in Barrett's esophagus with high-grade dysplasia as compared to normal. And if you look at the Barrett's progression from negative for dysplasia, indefinite dysplasia, to high-grade dysplasia and carcinoma, you see progressive overexpression of the line 1 protein. So this is something that had significant interest in our group for upper GI cancers more broadly. And then looking at it in gastric cancer in the beyond the cardia, we saw significant overexpression of the line 1 protein and the ORF1p expression in both signet cell diffuse type gastric cancer and intestinal type gastric cancer. So this looked to be potentially very promising for early detection of gastric cancer. So this was work that we published back in Cancer Discovery at the end of 2023, looking at this across various cancer types. And so we did really a pan-cancer analysis, and now we're sort of embarking on this with specificity into gastric cancer, because gastric cancer is one of the areas that appeared to be the most promising. And specifically, we partnered with David Walt, who is one of the leaders in terms of detection of various proteins in the blood using novel methods at the Weiss Institute at MIT, and what he's developed is a technology where you use ELISAs that are bound to various beads that you can use to amplify and detect, basically the ability to detect proteins in the blood at femtomolar concentrations. And so he developed this specific assay known as single molecule assays, or SIMOA assays. And then across all of our different cancer cohorts, we saw significant overexpression or high levels of this ORF1p protein in cancers, in particular ovarian, colorectal, and gastroesophageal cancer. Looking at that with respect to sensitivity, we also knew that we wanted to develop assays that potentially were more sensitive, in particular for early stage gastric cancer and early stage ovarian cancer. So David's group also developed a second generation assay, which was more sensitive. And I won't go through the details here, but essentially that second generation assay allowed for improved detection of the ORF1p protein in various cancers. And this specifically was something that we outlined in ovarian cancer. In addition, if you actually compare the detection of ORF1p with other blood-based biomarkers for ovarian cancer, we saw significant improvement in the ability to discern ovarian cancer patients from normal controls. And so this is just some ROC curves here where if you add ORF1p to other blood-based biomarkers, specifically CA125 and HG4, you see significant improvement in the ability to discern ovarian cancer from other cancers and also overall cancer from normal controls. And in particular, how does it work for gastric cancer? So we actually wanted to know, is it something that actually has specificity, particularly for early stage gastric cancer? So we also used another way of detecting the ORF1p protein through an assay known as Mosaic, which was also developed by the Walt lab, which allows potentially more sensitive detection of the protein using a little bit more plasma, in this case 500 microliters of plasma. And in this graphic, what we did was we took all the people who we knew had cancer but tested negative using the SIMOA assay and used the Mosaic assay. And the Mosaic assay was able to pick up gastric cancer significantly over healthy controls among people that were SIMOA negative. So this Mosaic assay is potentially much more sensitive and something that we really want to pursue more deeply. And you can see that in a separate cohort here where you see, again, higher levels of ORF1p protein among people with gastroesophageal cancer compared to those with normal healthy controls. And then finally, we wanted to assess the ability of the ORF1p protein to potentially be an early marker of response in patients with gastric cancer. And so what we did was in a gastroesophageal cancer cohort that was getting systemic therapy, we classified people as either responders or non-responders to systemic therapy. The red are non-responders and the black are responders. And what you see actually is when you look at the ORF1p proteins, pre-treatment and post-treatment, you actually see dramatic drops in the ORF1p plasma levels in the responders, whereas you saw no decrease or very little decrease in the level of ORF1P in the non-responder cohort. So it has potential as being a marker of early response to patients that are getting systemic therapy for gastroesophageal cancer. And this is some data looking at this with respect to imaging, where you see more substantial drops in ORF1P levels among those whose tumors are able to shrink on imaging. And we see quite clearly also significant differences if you just look at it from perspective of overall levels. The non-responder cohort here versus the responder cohort here, you see significant decreases in the ORF1P protein with treatment. And it appears to be prognostic. So when you compare people who have high levels of plasma ORF1P versus low levels of ORF1P, you see the levels that are low, those individuals tend to respond better to systemic therapy than to people who have high levels of ORF1P. So in summary, what we've been able to do with our stand-up team has been able to develop a very promising tumor-informed biomarker, specifically a plasma ORF1P SIMO assay, which appears to be very sensitive and specific to multiple cancers, really leveraging our knowledge of ORF1P as a potential important mechanism for the development of cancer. De-repression of ORF1P appears to be something that occurs universally across cancer, but in particular seems to be particularly relevant to gastroesophageal and colon cancer. And we're really working on multiple improvements to the assay. Specifically, we're trying to optimize this Mosaic platform to test this in a much larger cohort of samples. And so in the next phase of this work, with additional samples that we've collected in our Stand Up to Cancer International Biobank, we are going to apply Mosaic to several hundred patients to really understand the performance characteristics and then ultimately use this as a way to validate this as a potential biomarker that could be added for early detection. There are still areas that we really want to develop. In particular, we want to see also, is this protein potentially detectable in other body fluids, in particular the stool, because as we all know, the GI tract gives us an opportunity to sample specific production of various cellular products that are sloughed in the GI tract, so that provides an opportunity to potentially detect things in the stool. And this also is potentially an important tool for minimal disease, residual disease detection, as I showed in the last slide. And also this Mosaic and the Simoa technology is something that we can use and apply to other fluids such as stool and saliva. And then in other cancer types, we could also apply this as well. So this has been something that has been really of high promise and something we hope will move the needle, in particular for those areas of gastric cancer that are particularly challenging, like individuals that have hereditary diffuse gastric cancer. So with that, I want to recognize the team. This is the team over at MGH, but also I wanted to recognize the rest of our stand-up team, which we couldn't have done this work without. Certainly the international focus of gastric cancer has allowed us to bring together people from different countries. So we've really enjoyed our collaborations, in particular with Korea, where obviously we've heard a lot about the gastric cancer problem. And it's allowed us to really take advantage of the fact that we have so many patients in our Korean cohort that have early stage gastric cancer that allows us to really optimize our assays to detect early phase gastric cancer as opposed to later stage gastric cancer. So with that, I really thank you for the opportunity to provide this update and look forward to further discussion. Thank you.